Continuing on with our discussion of ecology, we'll now take a look at population ecology. And I have a few quick, simple terms to throw at you here. First of all, population, that's important. Population is the number of individuals of a particular species living in a specific area. And that area could be however you want to define it. If we're talking about humans, it could be a city, for instance. If we're talking about grizzly bears, it could be a provincial park, etc. Then we have population density. This is the number of individuals per area or per volume, especially if we're talking about the ocean or a lake. So for instance, we could be talking about the number of deer in a square kilometer. Dispersion refers to the pattern of the spacing of these individuals. Are they randomly dispersed? Are they clumped together, etc.? And then demographics is the study of vital population statistics and the study of how populations change over time. We'll start by talking about how we measure these things, and it can be rather tricky to measure populations, especially if we're talking about very abundant small organisms. If we're talking about large organisms, it might be possible to do a direct count. So maybe we can get some helicopters up in the air and go out and track caribou. We could perhaps count every single caribou in a provincial park. And this would be known as a direct count. So we get an absolute number. We get the number exactly right for that area. A human census would be an example of a direct count. People go out, they go door to door, and they count the number of people in each household, and also, of course, get some statistics about them. Maybe their income, their politics, etc., whatever it is you're interested in, but we can count every single person. Let's say we wanted to know exactly how many fish there were of a certain species in a small lake. So how many perch we have living in this lake. Now, most of the time, you would come up with some way to estimate that number. It's difficult to go out on a lake and look into the lake and see all the fish and count all the fish, of course. So usually we estimate, and we'll come back to how we do that. But there is one way, and it's a pretty drastic way, of counting those fish directly, something known as electrofishing. If you have a small pond that you're studying, you can literally zap those fish. You can run electricity through the water, kill the fish, and they float up to the surface. And you can count them. You can physically collect them and count them. Now again, that's kind of dramatic because you're killing the population that you're trying to study. Here we see another example of electrofishing, where we've got someone who's well insulated with rubber boots and waders, and they're wading through this small creek here and zapping the fish so that they can count them. Again, a rather extreme technique, and of course it's going to cause some harm to the environment. It will recover eventually, but if it's a case where you've got a study pond or a study stream and you need an exact count, in some cases it can be the best way to do it. Often there's just way too many individuals in your sample area. You can't possibly collect them all, or in the case of humans, talk to them all. So what you do is you take a small sample and you extrapolate from that. And here's a sample with humans. This is from our last election in Canada. What they're trying to do here is they're trying to figure out how people are going to vote in the upcoming federal election. Now, it would take a lot of work to send people out door to door every door in Canada and ask every single person how they're going to vote. So instead, what they usually do is they do polls over the telephone. And you can see that first poll there, if you look down the bottom, it says N equals 1,000. That's the number of people that they contacted, 1,000 people. And in the second poll here, we can see that they contacted a few more people. They contacted 2,651 people. Now again, that's a far cry from the 35 million or so people that live in Canada. But the more people we ask, of course, the better our estimate is going to be. So if you do have to just sample a small subset of individuals, which is the norm, you want to make your sample as large as is feasible. There are indirect ways of estimating population sizes, and some of them are pretty creative. So you could perhaps do what's called a drive-by or roadside analysis. So let's say you wanted to know how many bald eagles were found in a certain area. You might drive down a stretch of road, and maybe you decide that you're going to drive 30 kilometers down a stretch of road, and your passenger is going to be very eagle-eyed, so to speak. They're going to look out, and every time they see an eagle, they're going to tick that off on a piece of paper. So they're going to count just by eye every eagle that they see. Now, extrapolating this is difficult, but at the very least, you can get a sense of trends. So you could drive that same 30 kilometers at the same speed a week later and do the same thing, and maybe you do that every week, and at the very least, you're going to see, hopefully, if the population is increasing or decreasing. Similarly, ecologists, believe it or not, they will count roadkill as an indirect measure of the density of a particular population. So they'll drive down the road, they'll get out when they see a roadkill, they'll figure out the species, they'll mark that down, and once again, it's kind of difficult to extrapolate this to the larger area. There's a lot of things you have to consider. Of course, some species are more likely to get hit by cars than others. It doesn't work as well for birds as it might for raccoons, that kind of thing. But again, if you do this frequently, if you keep tabs on roadkills on a certain stretch of road, and you do that quite often, you should at least see trends. You'll see changes and maybe seasonal changes in population density. A rather common way of estimating the population of a species of animal is something known as mark recapture. Now what you do here is you go out and you catch, without harming, a sample of individuals, and then you tag them in some way. And then you let them go, and you let them disperse, and then maybe a few weeks later, a month later, you go out and you capture a bunch of those animals again, and you look to see if they have tags. And you can estimate from the percentage of individuals that you catch that second time with tags what the larger population is. Now there are some things that we have to think about when we use this. First of all, it can only be used on animals that are capable of dispersion. And you can't use this on things like plants because they're staying put. If you go out and tag plants in a forest, they're not going to disperse. So you have to have an animal that is motile, meaning it moves around and it can disperse itself. But we can do this with slow-moving animals as well, even something like a snail. We just need to give them more time to disperse. This technique is frequently used to estimate the number of fish in a lake. So let's say we have a large lake and we want to know how many fish of a certain species are in that lake. We can't electrify the whole lake, nor should we try. We don't want to kill everything in it. But what we can do is we can go to the lake and we can catch some fish and then put tags on them, so mark them, and then release them. And then go back later and catch some fish again and see what proportion of the fish we catch have the mark, have the tag. And we can extrapolate from that. So as an example, let's say we go to our lake and we catch 100 fish and we tag them all and we release them unharmed back into the lake. We come back after a month, let's say, and the fish have randomly redistributed themselves. And we catch 100 fish again. And 10 of those have the markers, have the tags that we applied before. That would imply that 10% of the fish in the entire lake were marked. So it would imply that there are 1,000 fish in the lake. The actual equation is here. So big M is the number of members of the population that were initially tagged. Big N is the population of the whole lake. That's what we're trying to estimate. And that ratio should be equal to the number of marked individuals we catch the second time, that's little m, over little n, which is the number of members of the population that we caught. And it doesn't have to be fish. It can be anything that will disperse itself randomly. So we could go out, we could catch mice in traps and mark them, and then go back a few weeks later and recapture them. Okay, but what do we do if we want to estimate the population of something that doesn't move around? Maybe plants, or maybe we're looking at a tidal zone and we're interested in the number of seaweeds or the number of mussels or oysters, something that can't move and redistribute itself. Of course, we can't use mark recapture. We might use something known as quadrat sampling. Not quadrant, quadrat. What you're seeing here is we have some ecology students, and they are in the intertidal zone. And they've stretched out a line here, a straight line. I'm not going to draw it too straight, but there's a straight line, a rope down there. And then every so often along that rope, they're going to lay down a square. And they're going to count everything they're interested in. So maybe they're looking at oysters. They would count all the oysters in that square. And they would do this at set intervals. So they might pick up that square, move it 2 meters further down the line, and then do their count again. From that, you can extrapolate. So let's say we have this 0.5 meter quadrat, that's our square, and we do this 20 times down the line. So we know in that case we have measured 10 square meters. And then we can estimate, maybe from a drone picture or something like that, what the total size of the area is that we're interested in. And maybe the area we're interested in is 1,000 times bigger. We can take our number from our quadrat analysis and multiply it by 1,000. And that will give us an estimate of how many oysters we have in that area. You can do this in forests if you're interested in measuring the number of plants, like maybe mosses or ferns or trees. We can even do this underwater, as you can see. Here's a similar technique, and here they're using a transect line. That's like the string that we saw in that last example. So you lay down a string or a rope or a measuring tape, and that would be this line along here. And then perhaps every 10 meters or so, you measure every single tree that's within 1 meter. So you measure out, and if a tree falls in to this 1 meter circle, then you count it. And you do this, you go down your line. Again, it might be every 10 meters, and then you stretch out a line 1 meter, and you check off every tree within that circle, and you do this down your line. And it means you don't have to count every single tree. So it makes your sampling much easier. And you might do several of these transect lines. They could all run parallel. They might be, well, 20, 50 meters apart. Again, it depends on what you're sampling, how big your plants are. Are you sampling trees or are you sampling little tiny moss plants? But you don't have to count every single tree, but you should get, and this is the key point here, a random sampling. So this is done to try to avoid any kind of bias. So you get your random sampling, and then you can extrapolate from that and try to figure out the actual number of members of the species you're interested in the entire study area. And you can do that to also see trends. Maybe you're doing this down a slope on the side of a mountain, and the climate, the microclimate, is going to change as you go down the mountain, and you might see species changing as you go down your transect. Or here we have a field, and then that grades into a forest. And of course, the plant species are going to change. So you might not just be looking at a single species. You might even have a few species that you're counting, and you can see their distribution patterns along these transects. So in these examples here, we've got 4 transect lines, and this time we're interested in estimating total populations, but also looking at trends, changes in plant species from the field into the woods. Ecologists are quite interested in patterns of dispersion. Now here's an example where we have a non-random pattern. If we look at the starfish here, we can see they're all clumped together. We have this clumped pattern. Now, why might that be? Well, in this case, it's probably due to the fact that these starfish are engaged in sex. So we have a little orgy going on here. But there can be other reasons. We can have some organisms that form little colonies. We can have organisms that live together that are social. So think of like ants, things like that. But how organisms are dispersed can tell us a lot about how they interact with each other within their species. And also it can tell us a lot about how they reproduce. If we're talking about plants, it can tell us about how effective they are at dispersing their seeds, for instance. Here we have some penguins, and you can see they are very uniformly distributed. They are pretty much the same distance from each of their neighbors. And the reason for this is territoriality. So some animals will maintain a territory. And here what we've got are a bunch of male penguins that are incubating eggs. So the female will lay an egg, the male penguin will take care of it, and right now they're plopped down on top of them, and they have their space. They defend this little bubble around them. So they're kind of social distancing, I guess. But they maintain this territory, and they will very aggressively fight off any other penguin that comes into their bubble. Here we're seeing a field of dandelions, and you can see that the distribution appears to be totally random. And that kind of makes sense. The dandelions, of course, aren't moving. They're not territorial. They're not defending a space, but they release their seeds into the wind, and the wind randomly distributes them. Now, we don't always see random distributions with plants. It's quite often based on the environment. So even in a field, we might have some areas that are a bit wetter. We might have little depressions that collect water. Or we might have hills that are drier, or perhaps the wind flows faster over the little hills, and that can affect seed dispersion. But the soil itself, whether or not it's wet or dry, how much nutrients, that can determine how well the plants survive there. So sometimes we do see patterns, but dandelions are pretty good at surviving most soil conditions, and typically the wind that disperses them in a random pattern. Population numbers do tend to change over time. If you remember back to when we talked about Darwin, he noted that populations stay roughly the same, and this is true if we're talking over decades, but from year to year and from season to season, populations do go up and down. They're dynamic. Now, over longer periods of time, they fluctuate up and down, but they maintain kind of an average, and we refer to that as a dynamic equilibrium. So what is responsible for changes in population numbers? Well, increases are the result of births and also the result of immigration. So we have individuals of that species migrating in from an outside population. And decreases are due to deaths and also due to emigration, individuals leaving the population. Again, these things may or may not always be balanced. Demographics or demography refers to the study of the vital statistics of a population and how they change over time. We might be talking about birth rates, death rates, reproductive rates. If we're talking about death rates, we employ something known as a life table. And a life table is the age-specific summaries of survival patterns of populations as we follow a cohort. Now, what's a cohort? Well, think of how we use this term every day. You might talk about your school cohort. So the people that you started your first year of college with, as you progress with them through your college experience, it's a cohort that travels along from year to year. When we're talking about these life tables, we're talking about individuals within a population that were born at the same time. So they started life as a cohort and they progressed together from year to year. Incidentally, life tables are used by insurance companies as well. They look at, how long we expect segments of the population to live, and they base your insurance premiums on that. Anyway, here we have a fairly typical life table, and this is for ground squirrels that are found in the Sierra Nevada of California. We're looking at one small isolated population here, and we're looking at a cohort. So we're looking at a group of squirrels that were all born in the same season. And you can see at the start of this study, we have 337 females and 349 males. By the second season, some of them have died. We only have 252 females and we have 248 males. So also on this chart here, we have our proportion alive at the start of the year, and you can see the death rate's pretty high during the first year. We had 125 females die and 140 males die. And our death rate there, you can see for females, is 0.50, so 50%. And our death rate for our males is 0.56, 56%. Now, as we go down from year to year to year, we look at these numbers here, you can see that the females, some of them, live much longer than the males. There's some females that survive into the 9th to 10th year, whereas all the males are gone at the end of the 6th year, the 5 to 6 years. And that's probably due to different dangers that they encounter. It might be due to the fact that males are defending territories, they're fighting each other. Being out there doing that makes them more visible to predators, for instance. Now, for our population of ground squirrels that we just looked at, we could plot the survivorship curve of the females and the survivorship curve of the male. So we're just plotting those numbers that we just saw in that table. And notice on the y-axis there, this is a logarithmic scale. And we see that the males die out quicker and they have a steeper decline, as you can see. When we start to compare survivorship curves between different species, so not just comparing sexes here, these are averaged for the sexes, we see some very distinct differences. So look at the one in the middle, the straight line down the middle that's labeled with the Roman numeral 2. That is for the ground squirrel. And we can see from year to year, so down the bottom there, we have the percentage of their maximum lifespan. We can see on a logarithmic scale that we get this straight line. So they do have a higher drop-off younger in life, but when we plot this on a logarithmic scale, we get a straight line. Now compare that to humans. Humans are the top one there, number 1. That survivorship curve is very different. In humans, we have a small number of offspring, children, and we are very protective of them and we take very good care of them. So we tend not to see a huge drop-off until we reach old age. And then, of course, we have that massive drop-off. Compare that to survivorship curve number 3. What we have here is an oyster. Now, what oysters do is they produce larvae that they dump into the water, and they produce tens of millions of them. So each female is going to dump millions of these larvae into the water, and the vast majority are not going to survive. Very, very few of them survive the early stages of development, but then once they finally settle down and become established, then they tend to fare pretty well. So we have this incredibly steep decline at the beginning, and then things level off. Reproductive rate has a major influence on population changes. And in studying the demographics of a population, we might also want to make a reproductive table. This is where we look at changes in reproductive rate as a cohort ages. Now, to do this, typically we'll only study females. That might seem a bit sexist, but females are responsible, of course, for producing offspring. In a lot of sexually reproducing organisms, the males aren't nearly as important. You don't need very many males to fertilize the females. Here in this table, we're looking at the same population of ground squirrels that we were talking about earlier, but this time we're focused on the females and their reproductive rates. So if we look in the first row here, our first column, of course, those females born in that first year, and again, we're looking at a cohort that was all born at the same time, and we're tracking them through the years. that first year in the life of that cohort, they were too young. They were not sexually mature. We didn't have any babies. We didn't have any offspring. In the second year, so the first to second year, we have some of those females maturing and finding mates and giving rise to litters of offspring. So you can see we have 65%, so 0.65 of our females in the second season having litters. And the litters are fairly small, so the mean size, this time including all the babies, males and female, is 3.3 babies. And the number of females is 1.65, so roughly half as we would expect. We're interested in the females because, of course, it's females that are going to give rise to litters of their own as they age. And then in the next season, 2 to 3, we have most of our females finding mates. They should be all mature at this point. And we have slightly larger litter sizes. And you can see the litter sizes kind of peak in that 4th to 5th year of age. And then after that, of course, they start to drop again, but the females at this point are quite successful at finding mates. So all of them are actually mating. Now, what about the actual growth of the population? Quite often, if there's nothing holding a population back, if there's no space limitations, if there's no limitations on food or other resources, they will grow at an exponential rate. This is especially true for microbes. So if we take bacteria, something like E. coli, they can reproduce every 20 minutes. So if we put one cell, one bacterium, into a flask of media that's got all the food it needs, that's like an entire ocean to that cell. After 20 minutes, it will divide into 2. Then after another 20 minutes, those 2 cells will divide, we'll have 4. And after another 20 minutes, we'll have 8, etc. And after 12 hours, as you can see here down the bottom of that table on the left, we have an incredible number of cells. Now, those cells will eventually get to a point where they're competing for resources. They've maybe finished up all that food. They're starting to release poisons, toxic waste products that will they'll poison each other with. But until we get to that point, we'll have this exponential growth. This sort of growth can happen in large animals too. Takes a bit longer, but we can still get this exponential growth occurring. So what you're seeing here is a population of elephants that was almost completely extinguished in an African park. So of course, elephants have been historically killed for their ivory, for their tusks. So in this park, the elephants were almost completely wiped out, and then some protection was granted them against poachers, and they slowly doubled their numbers. It happened slowly at first. Elephants have a long gestation period. It's quite a long time between generations, but the numbers went into this steep incline due to this exponential growth. If you remember, when we talked about Darwin, we talked about someone called Thomas Malthus. Not a name I expect you to remember, but he mentioned that what quite often happens in human populations is we might have this exponential growth that occurs when conditions are good, but eventually that exponential growth will outpace the ability of the population of farmers, in this case, to grow enough food. And then we get to a point where we have a crash. Now, he was noticing that societies got better and better at growing food, and that's what you're seeing in the bottom graph here, that the food production line is going up as more and more area is tilled. So as we have bigger farms and more food being grown, the population tends to go exponential. But then it hits a point where there's not enough food and it crashes. And then it does it again. It goes through these crashes. The number of individuals that an environment can support is known as the carrying capacity. And on graphs, this is symbolized as the letter K. So K is the maximum population of a species that the environment can sustain indefinitely. And as we'll see, there are different strategies for dealing with this carrying capacity. Some species will reproduce very quickly, exponentially, and they'll just shoot right through that carrying capacity, and then there's a huge crash, and they'll move on somewhere else. Whereas other species will approach this carrying capacity with a bit more care, and the population levels off, and it tends to maintain itself at a stable number. Without getting into the calculus of this, when we have exponential growth, we refer to the rate of change as little r. Exponential growth, again, is where we might have a doubling. So we have a population that doubles in size, and then it doubles again, and then it doubles again. So very, very quick growth. The blue line here shows this exponential growth. And organisms that behave this way, that keep dividing uncontrollably at this exponential rate, are referred to as R strategists. Now, also on this graph, we have a dotted line at 1500, and this is the K value for a population. That's the carrying capacity of the environment. That's the number of organisms that the environment can support. So for instance, if we're talking about herbivores, at this K value, the plants can grow fast enough to sustain a population of 1500. Our strategists don't care about the carrying capacity. So typically, they will move into an area where there's no competition. So they've got a brand new, fresh start somewhere. Let's say there's been a forest fire, it's wiped everything out. They move in, they colonize it, they grow incredibly fast. They grow right up through the carrying capacity. And of course, they're going to crash. But once that crash comes, they've already moved on to another area. Also on this graph, though, we have a red curve, and that is the growth of what's called a K strategist. Now, a K strategist, if it finds itself in a new area with no competition, it might grow exponentially at first, but then it will start to slow down. So it responds to changes in population density. It responds to changes in resources, and it starts to level off, and it tends to live right around that carrying capacity. K strategists, as we'll see, they tend to be organisms that produce a small number of offspring and take really good care of them. And because they produce a small number of offspring, their populations tend to grow much more slowly than our strategists. Now here we have a population of sea lions, and they were almost hunted to extinction, but then they were protected and their population started to rise. But when it got to the point that the environment was at its limits to support them, the population leveled off. This is known as logistic growth. And if it helps, you can remember it as kind of a logical growth. They're going to stay put in that environment. They're not going to do themselves in and move somewhere else. Instead, they're going to maintain their population at or around the carrying capacity. Now, that's not to say their population will go up and down. It will, but it will always average out at about the carrying capacity. And that's because their population changes fairly slowly. Let's have a look at what determines reproductive success. So how fast a population can increase in number. First of all, the age at which sexual maturity is reached. In invertebrates, it's usually very quickly. So for instance, with most insects, they might just be around for one season. But think about humans. It takes much, much longer for us to reach sexual maturity. How often does the organism reproduce? Just once or several times over the course of its life? And how many offspring are produced per reproductive episode? So let's take a look at our strategists. Our strategists tend to reach sexual maturity really quickly. They tend to, in many cases, just reproduce once, but they produce a lot of offspring. Now, that reproducing just once isn't always the case, but it is the case for, like I said, a lot of invertebrates. Let's take a look at the trade-offs of reproduction. So every offspring that's produced is going to require some energy. It takes energy to make the gametes, the sperm and the egg, especially the egg. A lot of energy has to go into making the eggs. Remember, they're much, much larger than the sperm. It takes energy to raise those offspring as well. And the more offspring you make, the less energy you can spend on each one. In many, many species, we have offspring that are produced in very large numbers, but the offspring are smaller and more vulnerable, and their chances of survival are small. But that's okay because there's just so many of them. So again, the two strategies, the R strategy, you can take the energy that you have available for your offspring and use it to make as many offspring as possible and then just dump them into the world and just hope that some of them survive. And it's going to be a very small number that survive, but, odds are in your favor that some will. Or with K strategists, you can take that energy that you have to spend on your offspring, only produce a small number of offspring, but then invest all that parental energy into raising them and taking care of them. So like I said, both strategies are potentially very effective. Now here's a couple terms I don't expect you to remember. They're not on your terminology list. But we have something known as semelparity, and this is where a whole bunch of offspring are produced all at once by organisms that only reproduce once in their lifetime. And then we have what's called heteroparity, which is where there's several reproductive events throughout life. So for instance, semelparity, think of an insect. Let's think of a praying mantis. Now, praying mantis will make an egg case, and it will contain thousands of eggs. And they'll all hatch out at the same time, and the mother just walks away and leaves them. But some of them are going to survive. Even though a huge number of them are going to die instantly, they're going to get eaten by ants, etc., some of them will survive. And heteroparity, this would be the ground squirrels we talked about. They reproduce every season. They have fairly small litters. And also humans, we reproduce, or we can reproduce several times throughout our lives. Here's a nice example of semelparity and a nice example of an R strategist. They're the ones that use this strategy. Big bang reproduction. You go out with a bang. So we have here some salmon. And of course, they migrate upstream from the ocean. They literally swim themselves to death, getting back to where they were raised as young fry, as young fish. At the top there, we have a female, and below that a male, the male actually morphs, it changes, its face becomes quite ugly as it swims upstream. But they will find a quiet little pool in a river somewhere, and they will lay their eggs. So the female will lay a big batch of eggs, and then the male will squirt sperm over top of that, and we have external fertilization, and then lots and lots and lots of little baby fish will grow up in that stream and eventually swim out to the ocean. Most of them, the vast majority, will not survive back to the ocean, but a small number will. And here we have a squirrel. It's going to have small litters several times through its life. And like I said, if we look at humans, that's an even more extreme example where we have just a small number of children, and we have those in separate events, usually throughout our lives. So to sum up, what's more important, quantity or quality? For our selection species, our strategists, it's quantity. They produce lots of offspring. The offspring are usually small. Quite often we're talking about a larval form of something. And they have a small chance of survival. But they can grow populations much more quickly. So if an R strategist finds itself in a place that's uncrowded, like I said, let's say there's a forest fire and it wipes out everything that was there. They've got a clean slate to start with. Put out as many offspring as you can, and you can increase your population quickly. Remember, evolution, that's what it's all about, trying to put as many successful offspring out there as you can. And then for K selection, so K strategists, it's quality of offspring. Typically we see this in a well-established ecosystem where there might already be a fair amount of competition and we're close to our carrying capacity. It's a high density situation, so you want to produce offspring that are of high quality to make sure that they're likely to survive. And this is a typical graph for R selected species and K selected species. You can see the K selected species climbs up to the carrying capacity and then kind of oscillates around that. So it is a bit dynamic. It climbs a bit too high and then goes down and a bit too high, but the average is going to be right on that carrying capacity. Whereas an R selected species goes through these boom and bust periods. Its population explodes, it uses up all its resources, it crashes, and then it explodes again, etc. On the bottom here, we're seeing these different animals put onto a range from R to K. So R, we have an oyster there that's producing 500 million offspring a year, and then we have a chimpanzee that's producing one every 5 years. One more interesting question, what's the best density? What's the best population density to have? Well, if your population is too high, there's going to be lots of competition, resources are going to be limited. If your population is too low, you're going to have a hard time finding mates. This is something we're going to talk about more in our next topic. This is a problem for individuals that are at the top of the food chain, top level carnivores. So for instance, a cougar. Cougar populations are fairly small because it takes a large number of rabbits and other prey to support them. And they can suffer quite a lot if their populations get too small because they have a hard time finding mates. Now, why do I have a picture of Sasquatch there? Well, one of the best pieces of evidence against Sasquatch is that if they do exist, they have to be remarkably rare. And how do they find mates if they're so rare? If they're so rare that we encounter them so infrequently, there's what, one every few hundred square kilometers? How are they surviving? But this issue, where in top level carnivores, we have so few individuals that they have a hard time finding mates is something known as the Ali effect, and we will come back to that in our next topic. Now, what about us? What about humans? Well, for most of our existence, we were at our carrying capacity. If we look at the ancestors of modern day humans, if we look at the earlier hominids, we were roughly around 1 million individuals in Africa. And we were restricted, like I said, by our carrying capacity. Now, after agriculture became a thing, we could create a lot more food. And then, of course, we developed sanitation and medicine, and our carrying capacity went through the roof. There's no shortage of, what we can do. If we look at the last 500 years, our population just exploded. So we have this very, very rapid exponential growth. And we were able to keep up with that. We were able to supply enough resources to support that exponential growth. It would appear, however, that human population is finally starting to level off. And it may be more due to cultural changes than it is to the carrying capacity. So in purple there, that purple line, that shows us the growth rate, the annual growth rate averaged out for humans around the world. And you can see it peaked in 1968 and it's sharply declining. In some countries now, the birth rate is less than the death rate. Now, whether or not the population of a country is going to go up or down depends on those 4 factors we talked about. So we have to look at immigration with an I and birth rate. And if that value is higher than the death rate and the emigration with an E, then we're going to get growth. But like I said, growth rates are going down in most countries. Some of the less developed countries are kind of lagging behind, but it's thought that by 2100, we will see a population leveling out at about 11 billion people. Right now, we're at about 8 billion people. We can estimate whether the population of a country is going to grow by looking at these tables that you see here. Now, this is not considering immigration and emigration, of course. But you can see for Afghanistan, for instance, we have a very wide base to this table. This table is showing different age cohorts as we go up. So we're starting with infants at the bottom, going up to elderly individuals. And you can see the base of this chart, of this bar chart for Afghanistan, is very wide, which suggests, of course, that the adult population is going to increase in the future. The United States has very slow growth. We have this kind of fat part to the curve here, and that would be the baby boomers. And I should mention this graph is kind of old. And then we have other countries like Italy, where we don't have any growth at all. And the reason for that is you can see we have a very narrow base. Species don't exist in a vacuum, of course. So if the population of one species changes, it's going to have an impact on other species in that ecosystem. Here's a classic example. You can see in blue fluctuations of snowshoe hares. And in red, you can see fluctuations in the population of lynx, one of the main predators of the snowshoe hare. You can see that if the snowshoe hare population increases, then the lynx population increases just a little bit behind it. It takes a little while to catch up. And then as the lynx population increases, that's going to have an impact on the snowshoe hare. It's going to crash. Then shortly after, the lynx is going to crash, etc. So population fluctuations between these two species are very closely intertwined. And of course, we'll talk about these interactions between species in more detail in our next topic. Thanks for listening. I'm hoping that was interesting. Sorry if my voice was a bit strange. I've got kind of a bad cold. But here is some terminology to leave you with, and we have one more topic to go.